Good afternoon, everyone. There's nothing quite like feeling the energy in a grad slam room. So if you can hear me, say hello. Thank you so much. I appreciate that. All right. Good afternoon, everyone. I'm very, very excited to be here today. For those of you who are new to this event, I like to refer to it as the Super Bowl of campuses without a Super Bowl, without a football team. So you will hear, you already seeing some signs of celebrations happening around the room, and today is all about two things: fun and communication. Nothing else. That's all we're gonna do. Um, Let me start by doing the land acknowledgement and then we'll continue. As we gather here in person on our campus in San Francisco, we want to acknowledge the Ramaytush Ohlone people who are the traditional custodians of this land. We pay our respects to the Ramaytush Ohlone elders, past, present, and future, who call this place, the land that UCSF sits upon, their home. We are proud to continue their tradition of coming together and growing as a community. We thank the Ramaytush Ohlone community for their stewardship and support, and we look forward to strengthening our ties as we continue our relationship of mutual respect and understanding. I'm Nick Abley. I have the honor of serving as Dean of the Division of Graduate Education and Postdoctoral Affairs, which is a mouthful. We say GEPA affectionately. So we're just GEPA. And I have the honor of serving as Dean. Grad Slam is part of a bigger event at UCSF. It is part of Grad Students Appreciation Week, a whole week just celebrating our grad students. And we do that really because our grad students are part of the engine of what makes research work at institutions like UCSF. So oftentimes they're the lowest folks on the ladder rung. But today and this week, you're flying high. And I hope that many of you have already participated in some events and you're beginning to feel appreciated. Yes? You better say yes. Thank you, Sydney. I appreciate that. Okay. Now, if you're a grad student, we do this every year, so I'm not breaking tradition. If you're a grad student, just raise your hand. Just wave your hand. Wave your hand. Keep them up. Keep them up. Keep them up. Now, if you're not a grad student, go give a high five to a grad student and tell them congratulations. I really expected to see more movement than that, but okay. That's all right. There are lots of seats down here in front. Come on in, for those of you who are trying to get in. We're just getting warmed up. We're just getting started. So today we celebrate 10 of UCSF's finest. And as I said before, we're going to have a little fun along the way. There's still seats up here, guys. Feel free to come on down. Don't be shy. Dr. Duncan doesn't bite, so don't be shy. Come on down. I think most of you will agree that in the current environment, finding moments of celebration and joy is probably a good thing. Yeah? No? No. Right? Yeah. You know, you spend most of your lives failing, right? Hey, truth is truth. Right? But what do you call a player, a baseball player who hits .33? What do you call them? Successful. Not just successful. Thank you. Rock star. Right? So you spend most of your time in lab failing, and you live for that one moment where synergy happens And when it happens, you sort of run up and down the corridor, making noises, celebrating. Today, today is all about success. Today is all about communication. And today is about enjoying, for the moment, this element of success. So here we go. Each of our finalists today, there are 10 of them. I see 9. I see one waiting in the corridors. There she is. She's a little nervous. Why don't you guys just stand up for a moment? Let's just acknowledge all of you. All of you. Now I'm not biased or anything. But I wanna just point out that there are 9 women standing over there. Just saying. Okay, I had nothing to do with this. I had nothing to do with the judging or anything, but 9 of them. So each of our finalists today has taken a real challenge. We have asked them to distill years of complex, specialized work into 3 minutes that just about anyone should be able to understand and care about. If you don't think that is challenging, just try it at home. You'll get it. At its core, communication, just lost my space. At its core, communication is about connection, about making ideas accessible, relevant, and meaningful to people who are outside of these walls. When we do that well, we're doing more than just passing along information. We're helping people to make sense of it and why it matters. Anybody know who Neil deGrasse Tyson is? Yeah, a few people, and especially a day like today. Have you seen the pictures from Artemis? Neil said, "The good thing about science is that it is true whether or not you believe in it." Love the guy, amazing. But for that truth to resonate, it has to be communicated in ways people can understand and connect with. That is especially important right now. Public trust in institutions, including the academy, is something that has to be continually earned. I want you to appreciate that. It has to be continually earned. When we can explain why our research matters in clear, concise, and for people beyond our circles, in human terms, we invite people in. And when we don't, we risk creating distance where there should be shared purpose. That's part of the reason that today is so important and why we will always do Grad Slam at UCSF. These students are bridging that gap, how to make rigorous science accessible, relevant, and compelling. Now, I'm just gonna say this. Last year, I think I saw her in here somewhere. Yes, I did. Last year, we went all the way so we have the local competition, and then we have the system-wide competition. We went all the way up the ladder and won. No pressure. Sophia is sitting with us today, and I have a little plaque in our suite that they're asking me to give back, and I don't want to. So no pressure on our contestants today, but that is your standard now. Okay, all right. Thank you to the finalists for stepping up to the challenge, for representing the strengths of our graduate programs, and for reminding us that great science deserves and requires great communication. With that, I am going to turn it over to your Assistant Dean, Deanne Duncan, who will serve as MC for today. So she gets to do all the hard work. I get to cheer you on, and I will come back. That sounds like a threat, doesn't it? I will come back at the end to help distribute the prizes. Good luck to everyone. Have fun. Don't forget, have fun. And I'll turn it over to Dr. Duncan.
Hi, hi, hi. How are we doing this afternoon? Look at me. Not sure what time of day it is. How are we feeling today? Now, okay, so you know what the rule is, right? Those of you who have been to the Grad Slam and I've emceed for the number of years, you all should know what the rule is. What's the rule? What? High energy. That's the rule. Keep the energy high. We have 10 fabulous finalists in the back. You know I have my script, so I got to go through my script. You know I go off script, so I will go off script. You know I got my jokes, so I will give you my jokes. And this year, the new thing that I'm introducing, along with the trivia, and I'm going to repeat myself when I get to it, is I'm going to be shaming if folks get the trivia questions wrong. Every year I do it, but I feel like I feel a little different with my energy today, so I'm not sure what I'm capable of, so beware when you start raising your hand when we get to the trivia, okay? So this afternoon, we are here to hear from our 10 of our close to 1,000 PhD students. You hear that? 1,000 PhD students we have here. PhD candidates entered the Grad Slam contest by submitting their talks on video, and a group of screening judges comprising of faculty, staff, and alums reviewed the videos to choose the 10 finalists. So this was deeply competitive. These finalists have all been working very hard to make their talks as engaging as possible for you. Oops. Of course, the prize money at stake does not hurt. For the competition, the grand prize is, thank you. See, look, you gotta get with the cues. $4,000. The second place prize is $2,000. And the third place prize is $1,000. All of which are taxable. Plus you, see, okay, y'all are getting the vibe. That's what I'm talking about. Plus you, the ones at home, because I know where the camera is, the live and remote audience will decide the People's Choice Award. So this prize is worth $750. So this will all get you something in San Francisco if you are a prize winner. You will have a few minutes to cast your vote online once all the finalists have given their presentation. So I'll cue you up, nothing for you to worry about right now. These finalists are not awarded the finalists who are not awarded one of the cash prizes will receive $100. So again, everybody going home with a little cash, right? That helps out a little way. The other way the in-person audience can participate today is the trivia contest, as I've already alluded to. Between each speaker, I will ask a question, and the live audience member who gets it correct will win a small prize. I know you're very anxious to hear all of the students' talks, but before we get started, we have to welcome and introduce our judges. So with that, judges, when I say your name, could you please stand as I introduce you? So Dan Burnell, Vice Chancellor of UCSF Community and Government Relations. Next, we have Dr. Utreya Day. Dr. Day is a postdoctoral scholar in the Department of Bioengineering and Therapeutic Sciences and the UCSF postdoc slam champ last year. Welcome back. Y'all remember his talk, right? Who were here? It was good. Dr. Maura Gunn. Dr. Gunn is the host of Tech Nation and Biotech Nation on NPR and professor of biotechnology at the University of San Francisco. Welcome. Next, we have Dr. Ainora Sawyer. Dr. Sawyer is the Executive Director of Innovation and Entrepreneurship at the UCSF Innovation Ventures and Associate Professor in the Department of Orthopedic Surgery. And lastly, we have Dr. Shannon Smith Bernadine, who is Associate Professor and Nurse Scientist within UCSF School of Nursing. She is also a UCSF nursing PhD alum and the UCSF Grad Slam champ in 2016, which is the first year. So we got some royalty up in here. So thank you all so much for being here and for doing the hard work of assessing our students as they come up. And so the judges will be looking for these basic qualities in the presentation. So the first is, did the presentation help you understand the research? Second, was the topic and its significance communicated using language appropriate to an educated but non-specialist audience? And lastly, did the presentation make you want to know more about the research? Did it spark curiosity within you? So each talk will be timed, and any presentation that goes over 3 minutes will be disqualified. Yeah, I know. By the way, we have an expert in the room. We have our official timer, who is our UCSF registrar, Doug Carlson. So he's up front. Just a little bit of notes about Doug. So Doug is an official timekeeper for Cal football and basketball games. He operated the game. I know, right? You're like, huh? I didn't know. He operated the game clock for the NCAA West Regionals at Chase Center in 2022, and he spots the downs and yards to go for the scoreboard operator at the 49ers, and he also did this at the Super Bowl this year. It's some skills, I'm gonna tell you that right now, to make sure that we on time. This costs money, this costs points, this costs championships. So you got a big job to do. So before we get to our finalists, because you know we gotta run through all the details, the housekeeping, the last thing is please silence your phones. We don't want anything to distract the students as they deliver their talks, so no talking, texting, or flash photography during the student presentations. So judges, are you ready? Finalists, are you ready? Everybody, are you ready? Let's go! So our first speaker is Sarah Wade. She is in the Neuroscience PhD program. Her faculty advisor is Dr. Anna Malowski, and the title of her talk is Birdwatching in the Brain, a field guide to neurons and their neighbors.
It's where I belong, down at the Pink Pony Club. I'm gonna keep on dancing at the Pink Pony Club.
I'm turning 30 this year, so naturally, I've taken up bird watching. And I've noticed something interesting. Certain trees attract certain birds. Oaks attract woodpeckers. Redwoods attract murrelets. The type of tree, it matters to these birds. I've been thinking about this a lot recently because in my lab, we study a different kind of forest. Your brain is organized the same way. Neurons belong to different subtypes, like oaks or redwoods, based on their structure and function. And just like certain trees attract certain birds, I'm wondering, could certain neuron subtypes attract certain cellular partners? But until recently, answering this question has been almost impossible. Traditional methods rely on looking at which cells are close to a neuron cell body, but neurons are not compact. A single neuron can stretch the entire length of your leg with branches in every direction. It would be like bird watching and just looking at the trunk of the tree instead of the branches where everything interesting is happening. So I've developed a tool to change this. It's called NeuroNotch. Using NeuroNotch, I can engineer a neuron subtype to express a protein tag on its surface. When another cell comes along and contacts that neuron, it recognizes the tag, activates an internal signal, and starts to glow bright red. So instead of just guessing which cells have contacted a neuron, now we can see them and analyze them. We can finally answer questions like, do certain neuron subtypes attract certain cellular partners? And we aren't just mapping the healthy brain. When brain cancer invades, it's like an invasive species that completely rewires the environment around it. It recruits healthy brain cells to feed it and help it spread. We are using NeuroNotch to profile exactly which cells the cancer is hijacking. Because if we understand the cells it relies on, we can start to disrupt the connection. Understanding the brain's ecosystem used to be like bird watching without binoculars. But with NeuroNotch, the forest is finally coming into focus. Thank you.
Thank you, Sarah. I'll be rethinking bird watching from now on. Thank you. Okay, now for our trivia contest. So for those of you who are viewing our event remotely, because I'm looking at you right now because I know where the camera is, like I said before, this will be like watching Jeopardy. What are the other games that are going on right now? Wheel of Fortune? What are the other ones? All those games, right? Where you get to yell at your TV and you don't get anything, right? It's the same thing for those at home. So in other words, you won't have any prizes, right? You just have the satisfaction of yelling at the TV, getting it right or getting it wrong, right? Nobody knows. But you'll hope that you'll have fun guessing the answers anyway. So today's theme is a slice of history. So get ready for some timely history topics. For our live audience, please raise your hand if you think you know the answer, and obviously no Googling the answer on your phone. We don't have audience mics today, so Dr. Shin-Yi Shea, one of our team members here in GEPA, will point to the first person that she sees who's raised their hand. And if that's you, say the answer loudly and then shout it out, and I'll repeat it so those at home can hear. And so once the question has been answered correctly, I'll tell you more fun facts in order to give our judges some time to evaluate the talk that they've just heard. So are we ready for our first trivia question? Yeah. So before we do that, hold on, let me say this. I don't know these questions, so I think they're actually quite hard, so I'm looking forward to seeing how you all do. Remember, the jokes might come and I might be shaming, so just be mindful as I shared. So the question is, Napoleon met his Waterloo in 1815. But Waterloo struck again in the 1974 Eurovision Song Contest. Whose Waterloo was it? ABBA. Mm-hmm. How you know that? Yes, it's ABBA. Some fun facts. ABBA's win with their song Waterloo at the Eurovision Song Contest on April 6, 1974 didn't just earn them a trophy, it launched them into global superstardom. Before that night, they were relatively unknown outside of Sweden. Afterward, they became one of the best-selling music acts in history. Judges, do we need to keep going? We can go to the next speaker? Okay, so that's the only related fact you're gonna get. Thank you for getting it right, because we gotta keep the ship running. So now for our second speaker. Jacqueline Williams, who is in the Biological and Medical Informatics PhD program. Her faculty advisor is Dr. Jill Hollenbach, and the title of her talk is Decoding the Body's Login System, the Missing Signal in MS.
There are many ways that security systems protect us, from deadbolts to high-tech surveillance. And today, I want to talk about the security system you carry within your body, your immune system, specifically through the lens of three-factor authentication. You know, when you need a username, password, and PIN to access an account. I study how the body uses similar strategies to fight disease. In multiple sclerosis, or MS, the body's security system attacks itself, unable to distinguish threat from healthy cell. A critical checkpoint lies with one type of guard, the natural killer cell, It uses a 3-factor check. Factor 1, username, is a receptor on the guard called KIR. Factor 2, password, is a matching receptor on surrounding cells. These receptors shake hands, providing clearance. And factor 3, PIN, is a protein fragment sitting between the two receptors. So to understand this process, I analyzed DNA from thousands of MS patients and healthy individuals, looking for the master login that protects against disease. And I found a trend. Healthy individuals more often have a specific username and matching password. What about the PIN? Using computational simulations, I identified a surprising candidate, Epstein-Barr virus, EBV. Most of us know EBV as the virus that causes mono, the kissing disease, and it's a major risk factor for MS. But my research suggests something unexpected. EBV, when paired with the right genetic username and password, may act as a security feature. It's the final signal telling the guard this cell is infected, sound the alarm. So when the check works, the immune system is secure. It clears the virus, no damage done. But if even one factor is missing, the login fails. The guard doesn't get the full signal and doesn't respond. The virus slips through and stays, disrupting the immune system. And eventually, that system begins attacking the body itself. Now, for decades, we've treated MS by broadly suppressing the immune system. But what if we could fix the login? My work points to a 3-factor combination: username, password, and viral PIN that may act as a protective code. I believe if we can understand and recreate that code, maybe we can start designing therapies that don't just suppress the immune system, but restore the body's natural ability to catch the missing signal. Thank you. Thank you.
So our next trivia question, we're the global health, oh, don't put it up yet, hold on. Okay, thank you. We're the global health folks. Y'all not here today? Okay, y'all are usually here. So the question is, which global health organization was established in 1948, marking a major milestone in international public health? This I know you know. Uh-huh. What's the WHO stand for? Because there are multiple the Who's. Okay, thank you. She gave me an attitude, y'all. Like, how dare I ask her what the WHO is? So, yes, congratulations. The WHO was established on today, April 7th, 1948. I know. That's like, where do they find these questions? A date now celebrated each year as World Health Day, highlighting a major global health issue annually. The WHO's creation marked the first time countries formally united under one organization to tackle global health challenges like infectious diseases, life expectancy, and healthcare across the worldwide scale. World Health Day 2026, observed today, calls on people everywhere to stand with science. Who said period? Exactly. You know. According to the WHO website, under the theme Together for Health, Stand with Science, this year's observance launches a year-long campaign celebrating the power of scientific collaboration to protect the health of people. I love it. Judges, may I move forward? Okay. Next up, our fourth speaker, Naja Mandalano Luna. Hello. Who is in the bioengineering PhD program? Her faculty advisor is Dr. Tamara Alliston, and the title of her talk is The Hidden Side of Bone Strength.
When we move, we take the stairs, we pick up groceries,
or we dance our way into a stage, our bones are listening. They can feel everything, and they quietly reshape themselves in response to it. Now think about you and your friend who are the same age, same height, same bone density according to your doctor. You're out having fun dancing, and you both trip and fall, but your friend gets up and you end up in a hospital with a broken hip. Well, on paper, your bones looked exactly the same, but deep inside, something different might be going on. And, well, in fact, the thing is, we've only relied on one single number for the last decade to describe bone health, bone density. But density is like trying to judge a building by how heavy it is. And what truly matters for bone strength isn't how much bone you have, but how well it's built. And it all starts with movement. We know exercise helps keep our bones strong, and it does it beyond density. But us scientists, we just don't know how it works. And that's where my research comes in. See, contrary to what you might think, bone is not a rock. It is not a static thing. It is very much alive. It contains all these cells that act like tiny engineers, and they sense the forces from your daily movement and decide where to repair and reinforce the structure. And I study this. I do it by using high-resolution imaging and molecular analysis to look deep inside the bone and track how strength is actually built over time. And what I've found is truly striking is the fact that exercise doesn't just build more bone. It in fact reorganizes the bone that you already have. It changes the way cells communicate. It improves not just the quantity of it, but the quality of it. So in other words, it changes the living parts of it. The things we just never thought to intuitively think counted as strength because we keep thinking that bone is a rock. So my research is actually the first step in measuring those living signals and factors so that we can detect fragility before your bones break. Because we don't just want to know why your friend is okay and you end up in the hospital, but rather to build technologies that can change that outcome so that more of us can take that fall and still be able to stand back up. Thank you.
So Naja, your research is gonna help me when I'm out outside lands, dancing and things like that, right? Dancing with the peoples. Okay, so our next trivia question. I don't know if you all are gonna get this one. I know, I'm shady. Let me read it. Okay, on April 10, 2019, scientists revealed the first ever image of what mysterious cosmic object? Oh, that's Ben. Ben got it. It's black hole. I underestimated y'all. So, some related facts. The image was produced by the Event Horizon Telescope. Has everyone seen Event Horizon? Yes? Yeah, there's a movie. Did you know it was a movie? Who said that? Ernesto? Dr. Rojas? Yeah, it has Lawrence Fishburne in it. Oh, okay. I just want to make sure. See, I'm spinning some facts up here now. So the Event Horizon Telescope is a global network of radio observatories that essentially tune Earth into a planet-sized telescope. Using this telescope, scientists obtain an image of the massive black hole at the center of the galaxy M87. There is a supermassive black hole at the center of our own Milky Way galaxy, and Event Horizon captured the first image of that object in 2022, so this is recent. Black holes thought to result from the collapse of massive stars are extraordinary cosmic objects with enormous masses, but extremely compact sizes. The presence of these objects affect their environment in extreme ways, warping space-time and superheating any surrounding material. Did everybody know that? No, I know you didn't. Judges, may I proceed with our next speaker? Okay, our next speaker is Christine Boutros. She is in the neuroscience PhD program and medical scientist training program. Her faculty advisor is Dr. Michael Wilson, and the title of her talk is The Fungus Among Us, Fishing for Answers.
In 2023, I could not escape The Last of Us. Billboards, Reddit threads, group chats, everyone was talking about HBO's new show with a brain-invading fungus that turns people into zombies. And I just wanted to be able to come home from lab and not think about work. That was proving to be pretty difficult because my work involves a fungus that invades the brain. It doesn't turn people into zombies, thank goodness, but it can kill people. And for those that survive, it completely changes their lives. Part of the reason why I think a show like The Last of Us really captured the world's attention is that we tend to associate our brains with our sense of self. So when something threatens the brain, it feels like an existential threat. Now, the disease I study is called valley fever, and for most, it starts as a lung infection, but in some, it spreads to the brain where it causes inflammation to the brain's lining, what we call meningitis. So here's the mystery. Why do some people get the severe brain-invading form of this disease while others clear their lung infection and go on to have lifelong immunity? It's a bit like the lead character, Ellie, in The Last of Us. For reasons no one quite understands, she is completely immune to the zombie-inducing fungus. So what is her immune system doing differently? That's the real-life question I get to ask. To get some insight into this, we have to look inside the immune system. When people clear valley fever, their immune system leaves behind a record, what we call antibodies. Antibodies are proteins that recognize and remember invaders. If I can understand which antibodies are uniquely present in the people who clear disease, I can start to understand part of what protection looks like. I bioengineer thousands of tiny viruses, all wearing little bits of the valley fever fungus, kind of like baiting a fishing lure. And then I cast my line that's strung with thousands of these lures into the patient's blood. Antibodies will then bite or bind whatever they recognize. And by determining which lures get bit, I can read the immune system's memory. So far, my research has found that the people who clear disease have a much more broad set of antibodies than those who don't. The next step is understanding which of these antibodies are capable of clearing the fungus. As we understand this, we get closer to better diagnostics, better treatment, and maybe even better prevention. Because unlike television, this brain-invading fungus is real. And understanding immunity can be the difference between survival and slowly losing our minds to this disease. Thank you.
Christine, I knew this was connected, the HBO show and your talk, when I read your title. So you were spot on. So now our next trivia question is, which iconic cream-filled snack was invented on April 6, 1930? Well, what do you mean, huh? Yes, it's Twinkies. So if I may ask, when was the last time somebody had Twinkies in here? Who had Twinkie like in the last month? How about the last year? Nobody's hand is up? I know y'all have had Twinkies. I have not, so that is why legit my hand is not up. But I know somebody in this room has had Twinkies. They might be in the vending machine. So Twinkies were invented on April 6, 1930 by Canadian-born baker James Dewar for the Continental Baking Company. They're originally filled with banana cream. This I did not know. Right, exactly. But during World War II, bananas became scarce, so the company switched the now classic vanilla cream filling we know today. In 2012, Time Magazine ranked the Twinkie #1 in its list of 10 iconic junk food. That's why I asked the question. That's why I was like, y'all not telling the truth. hands need to go up, saying that they've been a staple of our popular culture and above all in our hearts. Often criticized for its lack of any nutritional value whatsoever, the Twinkie has managed to persevere as a cultural and gastronomical icon. Look, you're right, icon? Okay. Speaking of perseverance, despite the urban myth that Twinkies last forever, y'all need to listen to this. Their official shelf life is just 45 days. So if you ate something after 60 days, nevertheless, the world's oldest known Twinkie turned 50 years old in January. A long-running science experiment at Blue Hill School in Maine. The teacher who started the experiment back in 1976 now says, quote, "The Twinkie and I are a lot alike. We're old, gray, and flaky." I had to read all of those because that one was good. Okay, judges, are we ready to proceed? Okay, our next speaker has some coordinated folk. They got outfits coordinated up in here. I haven't seen the outfit coordinated. So our next speaker is Brandon Courteau. He is in the Tetrad PhD program. His faculty advisors are Drs. Natalia Ura and Clement Verba. And the title of his talk is Protein Love Island, How Recoupling HER3 Could Reveal New Possibilities for Breast Cancer Therapeutics.
I came here for love. I came here for love. I came here for love.
Welcome to Protein Love Island. On the surface of every cell in your body, there are special proteins, we call them receptors, looking for love. Their job is to find a partner, and when they do, they strike up a conversation that tells the cell what to do. Now, most of these relationships are healthy, but there's one particular couple, HER2 and HER3, that are quite the chatty couple. While they're normally very good at telling your cells to grow and divide, sometimes they get a little carried away, forming too often or staying together too long, and get stuck sending these non-stop grow and divide messages. This overexpression or overactivation is exactly what happens in cancer, particularly breast cancer. HER2 and HER3 become quite the dangerous power couple. Now, in cancer, we can fight this. Many cancer drugs work by breaking up these bad couples, or at least stopping their conversations. And oftentimes, that works, at least at first. One of the big problems in cancer treatment is that what works early doesn't always work forever. Over time, cancer can find new ways to keep the conversation going, whether that's by resisting the breakup, or when we break up one couple, cancer starts shipping another. We call this acquired or adaptive resistance, and it's a big challenge in cancer drug development. So what can we do? Well, that's where a surprise new bombshell enters the villa. In my work, we've made the surprising discovery that HER3 can also pair up with another HER3, which was not a recoupling we were expecting. So what's going on? To figure that out, I'm using a technique called cryogenic electron microscopy, or cryo-EM. In short, I'm rapidly freezing these proteins in place and taking millions of tiny atomic level pictures to build a 3D model of how they fit together. It's like hidden cameras catching them mid-chat. Well, why do we care what HER3 looks like? If we can figure out what this looks like, we can figure out what it's doing, whether that's helping cancer cells or a way to fight against them. Once we know that, we can use their structure to design new cancer drugs. Drugs that break up HER3, or maybe HER3 is in a healthier couple on its own, away from HER2, when we want to support HER3 in closing off with itself. Either way, HER3 represents a brand new, never-before-explored platform for cancer therapeutics. In the long run, this can help us design new therapies that make it harder for cancer cells to escape treatment. So, while everyone's been watching the main power couple, my project focuses on a more unexpected recoupling. Because in biology, just like in life, you might find love, or your next cancer drug target, where you least expect it. Thank you.
Did we get a picture of the color coordinated group? Can we get a picture of this? It's rare that y'all come in outfits like this. And I want to say, Brandon, I've known Brandon for almost a decade before he started here at UCSF, and you haven't missed a beat. This was you 10 years ago, so I love it. So our next trivia question. The California Street Cable Railroad Company began service on April 10th, 1878. How many passengers did it have on its first day? Ooh, here. Who's that? 11,000? You right. 11,000. So the California Street Cable Railroad Company kicked off service on April 10th, 1878, becoming one of the first cable car lines in San Francisco back when horses and steam were still common city transport. On its very first day, the line carried a surprisingly large number of passengers for the era, showing just how eager San Francisco's were to try the new gravity-defying cable cars that could climb the city's steep hills. Can I tell you, I get really scared going up and down the hills because I think it's a roller coaster. California Cable remained a profitable, independent company well into the 20th century, long after most of the other lines were only a faint memory. In 1952, it became an official part of SF Muni. Today, San Francisco's cable cars are one of the only 2 street railways designated National Historic Landmarks. Judges, able to move forward? Okay. Let's welcome our 7th speaker, Mika Matera Vatnik. Mika is in the Developmental and Stem Cell Biology program and Medical Sciences training program. Her faculty advisor is Dr. Peter Turnbaugh, and the title of her talk is The Hidden Wiring of Hunger.
Maybe you can drive my car, and maybe I love you. Beep, 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 yeah.
I was shadowing at the hospital when I met 2 patients with obesity who had just started taking a miracle drug for weight loss called Ozempic. 3 months later, the first patient lost an amazing 15% of her body weight. But for the second patient, the scale hadn't moved. This isn't unusual. Ozempic only works for 2 out of 3 people. And the problem is, we don't know why. So how does Ozempic work? Most importantly, it decreases your hunger. If we think about your body as a car, when fuel runs low, or when you're feeling very hungry, there's that little red low fuel light that turns on, telling you it's time to fuel up. Ozempic delays when that low fuel signal turns on. And what's still unclear is what exactly controls that signal, and if that is the difference between our 2 patients. That is what my research aims to uncover. So what could be controlling our low fuel signal? One overlooked suspect are the trillions of bacteria that live in our intestines, called the gut microbiome. Some of these bacteria can make hormones and signals that we already know can talk to our brain and make us feel hungry. So, does the gut microbiome even matter for how well Ozempic works? I designed a mouse experiment to test just that. I gave Ozempic to obese mice with and without a gut microbiome. I then tracked their weight and how much they ate every day for a month. Mice without a microbiome, when I gave them Ozempic, ate very little and lost a lot of weight. Unexpectedly, mice with a microbiome didn't lose as much weight. And in between doses, they ate more. That told me something very important. The gut microbiome isn't just along for the ride. It can actually turn on the low fuel hunger signal. If we can identify the specific microbes that can turn on our low fuel light and identify what that signal actually is, then we can map the circuit. Because current treatments don't work for everyone. Delaying the inevitable low fuel light isn't enough. We must understand and leverage the wiring underneath. Thank you.
Thank you, Mika. I like your image. I identify with the creature in the left bottom corner with the one eye. Are we ready for our next trivia question? So you all know the drill. You know I'm gonna stop and pause. Do you all think that was an acceptable response to me? Are we ready for the next trivia question? Thank you. So the question is, on April 9th, 1912, which governing body voted to consolidate the University of California Medical Department in San Francisco, helping form what would later become the University of California, San Francisco? Nobody's hands up? Y'all should know this. Okay. No, boo. Yeah. I was wondering if I needed to say the whole name. It's UC Regents, yes. So yes. So before the consolidation, the University of California had medical programs in multiple locations, but centralizing in San Francisco allowed the school to partner with hospitals and focus on hands-on clinical training, setting a new standard for medical education in California. In 1964, UCSF was given full administrative independence, becoming the 9th campus in the UC system and the only one devoted exclusively to the health sciences. The 26-member UC Board of Regents continues to be the UC governing body, which has authority over university policies, financial affairs, tuition, and fees. The board also appoints the UC president as well as the student regent, who serves a one-year term. So if this is something that you're interested in, we are happy to nominate you. Follow up with me afterwards, okay? Seriously, our office manages this. So if you want to do this, let us know. Judges? Okay. Our ace speaker is Vanna Tran. Vanna is in the Tetrad PhD program. Her faculty advisor is Dr. Sophie DeMont, and the title of her talk is Poking, Prodding, Pushing, and Pulling Cell Division Addition.
Tonight, but you've been so out of touch, and I brought you way too much, over all I think it is.
Do you remember playing with Play-Doh as a kid? Or an adult? Taking that blob and pushing and pulling it just right to get the kind of shape you want? Your body did work to generate force. It gets transmitted through your fingertips to the Play-Doh. And if you decide to split it in two so that your friend can play too, you pull and divide that blob. Well, cell division is basically just like that, except the Play-Doh is your DNA, the instructions that make you you, and your fingers pull on these rope-like structures called microtubules. The goal is to evenly divide your DNA into two daughter cells, not to give one half to your friend. And this is super important because cell division is the basis of multicellular life. None of us would exist without it. We all had our cells divide like trillions of times. Just listen to Grad Slam today. But integral to this process are kinetochores. It's a protein structure built on the DNA and connects to those microtubule ropes. It's like the middleman, transmitting force to our DNA in order to move it. I'm interested in understanding how our kinetochores handle large forces during cell division and how it keeps itself together. If you just left Play-Doh out, it can dry out and break apart when you play with it. If a kinetochore breaks, we get division errors, leading to a host of diseases like cancer. We know if you're poking and prodding Play-Doh, the material responds and changes shape because of that. Similarly, I use the microscope to look closely at our kinetochores under normal and even larger forces to learn about how it changes shape. Here,
I found that kinetochores are actually elastic materials, like rubber bands, able to stretch and then relax. This way, it doesn't break apart under large forces. But when I make the DNA softer, kinetochores deform much more. It can even split into multiple blobs. They're also more likely to let go of their ropes, and that is not a good thing. You won't be able to move your DNA without those ropes, and genetic information gets lost. This tells me that the underlying DNA foundation has to have just the right amount of stiffness and flexibility to help the kinetochore hold things together, but also have adaptability for changes in force in our cells. With this research, I found that our kinetochores and DNA are specifically tuned to have the perfect properties in handling large forces during cell division. Ensuring the fidelity of this process is essential for us to develop better therapeutics to target the cells with division errors while we allow the healthy ones to persist. And just maybe, we can glean some tips from these cells on how to evenly divide our Play-Doh so that your friend can poke and prod it too. Thank you.
And we have 3 sets of posters, right? Did we get all 3 sets? 4 sets? 3 sets, right? We got to make sure we get some pictures. Great job, Vanna. I'm just gonna have him take these pictures. We got to capture this. I really like it. Thank you. So are we ready for our next trivia question? Thank you. I appreciate you all. Is this the last one? No, we have one more after this. So, which baseball legend hit his 715th career home run on April 8th, 1974, breaking a long-standing record held by Babe Ruth? Joey, now you, Joey.
Okay, 3, 2, 1.
Yes, Hank Aaron. On April 8th, 1974, Hank Aaron of the Atlanta Braves hits his 715th career home run at Atlanta Fulton County Stadium, surpassing Babe Ruth's legendary record of 714 and cementing his place as one of the baseball's all-time greats. The thing that I remember most about that night was my mother, Aaron said in a 22 interview with Dan Patrick. She was so enthused, she was so happy. When I hit the home run and got around the bases to home plate, she grabbed me, and I had never been hugged before in my life like that. I know, right? See, y'all are getting it. Thank you. He would eventually amass 755 career home runs before retiring in 1976. Aaron held the record for 33 years until Barry Bonds, here in the Bay, passed him in 2007. Although the taint of the steroid era in baseball leaves Aaron in many people's minds as baseball's last legitimate home run champion. Judges. All right. Our nice speaker, Rafaela Shima. She is in the Tetrad PhD program. Her faculty advisor is Dr. Peter Turnbaugh, and the title of her talk is Quieting Molecular Gossip, How Ketone Esters Reshape Microbial Metabolism.
Começou a putaria aqui no baile de favela. Vai sarrando todas elas, vai sarrando, beleza, vai sarrando.
Most of us have been in a friend group where gossip starts to spread. At first, it seems small, a comment here, a rumor there. But if the environment is stressful, competitive, unstable, gossip spreads faster. And the faster gossip spreads, the more toxic the environment becomes. It's a feedback loop. Now imagine that this friend group are the trillions of bacteria living in your gut, the gut microbiome. And the gossip is a small but highly reactive molecule called methyl glyoxal, Mg, molecular gossip. Every time we break down sugar for energy, a little bit of Mg is produced. In small amounts, it's inevitable but manageable.
But in metabolic diseases like diabetes, MG accumulates and leads to long-term damage. Now here's where the system gets even more complex. It's not just our own cells that can produce MG, but the gut microbiome, the friend group, can produce MG too. And now the loop worsens. A metabolically stressed host promotes an environment that produces MG, and more MG further damages the host. It's a feedback loop. They amplify each other. In my PhD, I asked, what if we can interrupt this loop without dismantling the whole friend group? Using genetic sequencing, I looked at not only what bacteria were present, but also what genes they carried. In other words, what behaviors these friends in the friend group have. And what I found was striking. Ketone esters, this dietary supplement that mimics a ketogenic diet and has been shown to improve metabolic health without the extreme dietary restriction, not only maintained the friend group the same, but quieted the molecular gossip, quiet MG. And not only that, but improved glucose control. At first, we thought this was only happening in the host, but through this study, we found that they were also changing the genetic status of the microbiome. We then engineered out DHAK, this microbial gene, saw that it reduced MG, the molecular gossip, and saw that it was sufficient to improve glucose control in the mice and the humans. So ketone esters act through two ways. They not only reshape host metabolism, but they improve gut microbial health. Not who is there, but what they're doing. Finally, sometimes improving metabolic health is about breaking this feedback loop, not only calming the environment, but also quieting the gossip. Thank you.
I like your image too, Rafaela. It's very creative and colorful. I like it. All right, we are at our, let me make sure, our final trivia question. So, the question is, on April 7th, 1827, British inventor John Walker sold the first of these products that advanced fire starting technology. What are they? I didn't think y'all, yeah. I see some hands. You right. I need you to say it with conviction and confidence, even though I said I was gonna shame. It's matches. It's friction matches, but matches is fine. I don't know what friction matches are, but anyways, it's okay. Walker accidentally invented friction matches, which stirring chemicals when the dried mixture on a stick caught fire from scraping against his earth. He realized that he created a quick, portable way to start a flame. He called them friction lights and sold them at his pharmacy in Stockton-on-Tees. His first friction matches were made of cardboard, but he soon began to use wooden splints cut by hand. Later, he packaged the matches in a cardboard box equipped with a piece of sandpaper for striking. He was advised to patent his matches, but chose not to, and as a result, Samuel Jones of London copied his idea and launched his own Lucifers in 1929, an exact copy of Walker's friction lights. Learn something new every day, y'all. Did y'all know this? Yeah, okay, good. I'm gonna make sure I'm not the only one. Okay, our final speaker. Yes, go, Lexi. Our final speaker is Lexi Snyder. Okay, we got signs over here. Lexi is in the Biomedical Sciences PhD program. Her faculty advisors are Andrew Yang and Jason Sister, and the title of her talk is In the Spotlight, Illuminating Brain Therapeutics with Torch.
This year, I did something reckless. I decided to join the cast of a musical.
Now, I am not a singer, dancer, or actress. So instead, I'm trying to manifest success. And I keep imagining those final bows. Blinding lights, deafening applause, and a never-ending sea of faces. In that moment, I want to spot my friends and family cheering wildly in the crowd. But in all that chaos, how do I even know where to look? As a scientist, I actually face a similar challenge. I'm trying to direct drugs to specific cells in the brain to treat things like tumors or Alzheimer's disease. But the brain is like that theater audience multiplied by billions. It's a dense, complicated crowd of cells and proteins. To get a drug to the right cells, we need specific molecular directions, like a row and seat number. With that, our drug can find its way through the crowd, treat the sick target cells, and leave everything else alone. So, how do we give a drug directions? Well, on the outside of any cell live highly accessible surface proteins that a drug passing by could stick to. The challenge is figuring out which surface proteins uniquely belong to our target cell and not to a neighbor. In the dense crowd of the brain, it's a nearly impossible task. So, to help us see in the dark, we created a tool called Torch. Torch expands on a simple idea: using an enzyme, a tiny biological machine, to attach chemical tags to nearby proteins. But, well, if we put this enzyme on the surface of a cell, we could then easily collect and identify the surface proteins that it tags, giving us a short list of promising new drug targets. But existing enzymes weren't efficient for tagging these surface proteins in the brain. So we dug through the literature, found a new enzyme called tyrosinase, and spent several years testing and redesigning it. Now, using genetic instructions, we can program any cell in the brain to seat our enhanced enzyme, Torch, on its cell surface, and it will attach glowing tags to every surface protein within its reach. It's kind of like having a friend in the audience to tag my family with bright glowing hats. Suddenly, they're impossible for me to miss. We're now using our tool to map the surfaces of different brain cells, and it's helping us discover new drug targets to precisely shuttle drugs to cells in the brain. So while my parents might still be hard to spot in the crowd, with Torch, hopefully new brain targets won't be. Thank you.
Thank you, Lexi. I liked your analogy. That's quite brilliant, the research that you're doing, so thank you. So now I would like to invite all of our finalists back up to the stage. Let's give them a big round of applause. Let's go with the energy. I feel like we need a drone in here. Cause like we needed to capture like all of this like at once. So thank you all for educating us and entertaining us today. Again, let's give them a huge round of applause because it's not easy work. Congratulations, you can return to your seats. Thank you. So let me give you some instructions for the People's Choice Award. So this is our audience's chance, so audience at home, to play judge today. So vote once for your favorite talk to win the People's Choice Award by going to the URL or QR code that will be displayed on the screen momentarily. Those of you watching remotely will also see this link right under the video in your browser or in the description area below the video on the YouTube, so you may need to refresh so this comes up. So audience voting will be open for just 5 minutes. Everybody has their card in here? Everybody has their voting card, right? The voting card. Oh, we don't do it like that anymore? Okay. I'm old. I'm going to old school. I'm sorry, y'all, because we used to like detach it. My fault, because everybody's like, what? So it'll be open for just 5 minutes, and remember, just 1 vote per person. And so the judges, we're gonna go off into a room, Genentech and 114, and we will come back, I would say like 15-ish minutes. So just hang around here. The refreshments are outside. Just kind of hang out, see your peoples, get some hugs, get some pictures. Thank you all.
So thank you all for your patience. Welcome back. I think, as you might imagine, the judging was tough, but somebody had to make a decision, not named Dr. B. So want to thank the judges again for the hard work that they did in trying to identify 3 different winners. Thank you for your votes for the People's Choice. And I really just want to congratulate the speakers again, all 10 of you. My goodness, isn't it awesome to be at UCSF? You guys are phenomenal. All right, now I always forget this, so in my script, I put it first so that I won't forget, because it's important to say thank you. So first of all, what an amazing MC you had today. All right, so thank you, Dr. Duncan, for the hard work and for helping us to keep our energy up. Appreciate that. Grad Slam, you heard Dr. Duncan mention earlier about the coaching that goes on, the judging that goes on, all of this stuff. So we had about 5 judges. Some of them are in the room. I'm not supposed to call them out, but okay, I won't call them out. Some of them are in the room, but we have over 50 applications every year, and we have to whittle that down to 10 folks. So please thank all the judges who did the pre-screen and the final screens. David Blanche, I don't know that I saw him here today. David from OCPD works with them, most of them, all of them, to refine their talks. That is not an easy task to do. 3 minutes, you go home and try it. 3 minutes with all your work. So thank you to David for his hard work and dedication in helping all our finalists. Also want to say thank you to the ETS team. And once again, you don't know they exist until something goes wrong. And nothing ever goes wrong with this group. So thank you to the gents for the ETS team. This event does not happen without a lot of work from both our communications team and our events team. They pay attention to every single detail, down to the things that I will say. I always go off script, so it doesn't help them, but down to everything that we say, all the trivia work that you answered, some of the stuff that most of us didn't know, all of the, have you seen the stuff on social media? Probably haven't. All the announcements on social media, our comms team and our events team, they are truly amazing. I don't see Annalisa in here. Am I missing her? I don't see her in here. But if you see her outside, just tell her well done. Give her a tap on the shoulder. Tell her well done. She's amazing. Please give a round of applause to our comms team and our. And then lastly, I just want to thank Sam for his and the Chi Foundation for their contributions to Grad Slam. They're the ones that provide the dinero for the first prize winner. So, and Sam will get a chance to present the first prize winner in a few moments. All right, I'm done talking. Are you ready? All right. Thank you, Dan. I'm going to have Dan give a short announcement.
Sure. Thank you, Dean Blake. Hello, everyone. So I have some exciting news to report, and this is actually Dean Blake's idea, so thank you, Dean Blake. when the top candidate from each UC campus goes to Sacramento on April 22nd for the statewide grad slam, they are all going to be putting on their advocacy hats and serving as lobbyists for the state research bond that, if passed by the voters in November, would provide $23 billion to fund medical research. This is targeted mainly on research that has been disrupted or become uncertain from the federal government. So whoever is our top vote getter this time, they'll get a crash course in lobbying from community and government relations, and then we'll take you around. And the most important thing is that you bring your brilliance, your enthusiasm, and your passion for your research, and let our elected officials know how critical it is that your work is supported. So we're very excited. We're looking forward to seeing who our top candidate is here from UCSF, and my department will be taking you around to meet with some elected officials. So thank you, and it'll be really exciting. So thank you, Dean Blake, all her idea.
Yeah, you gotta learn to top the politicians too. Just a thing to do. All right, we have 4 awards to present today. The first award for $750 goes to, oh wait, no, I'm not doing that, sorry. I am going to ask Maura to come up and present the People's Award. I'm not doing that.
Thank you so much. And I have to say, as a judge, it's really harrowing in there. We did have a couple of, interactions about those things. But what do judges know? It's the voice of the people. Let's hear it for the People's Choice Award. Just had to get that straight. Jacqueline Williams. Oh, I was just Never miss a microphone.
Oh, okay.
Say thank you.
Thank you, everyone.
All right, for our third place winner, we're gonna ask our young postdoc, Atria, to present the third place.
All right, the third place prize goes to Naja Michelle Maldonado Luna.
And Shannon will do the second place winner. And you can open that and reveal.
I at least know the answer already ahead of time. And I have to admit, I'm almost just as nervous up here now as I was 10 years ago at the beginning. So I would love to introduce our second prize winner, Christine Boutros.
It's his wife. Always good to have you in the room, Ernesto. And now we're going to ask Sam to come to the stage, and Sam will reveal the 2026 Grand Slam winner. Some drum rolls? Yeah.
Thank you. The first prize winner is Brandon Chateau.
Sam. That check will not cash at the bank, but magically it will appear in your account somewhere, sometime. All right, we're gonna ask all the prize money winners, because you're all winners, to come down to take pictures, or maybe outside and take pictures, I don't know. The camera guy will tell you, to take some pictures so that we can record this for historical purposes. And he is very excited about that $4,000 check. You can't see him, but he's very excited about it. Down here. Okay, so down here, right after. Folks, thank you so much for being such a lovely audience. I hope you had some fun today. And we will see you for the rest of this week, for the rest of Grad Student Appreciation Week. Thank you all.